Hello, this is Dr. Steve Feldman, Professor of Dermatology at the Wake Forest Baptist Medical Center here in Winston-Salem, North Carolina. I'll be discussing the publication, the PROSIT-BIO Cohort, a prospective observational study of patients with inflammatory bowel disease treated with Infliximab Biosimilar by Dr. Fiorino and colleagues. This study was published in Inflammatory Bowel Diseases. I selected this article to discuss because of the large cohort of patients in multi-center study utilizing CTP13, the first Infliximab biosimilar evaluated by the European Medicines Agency, the EMA. Previously, little information was available on the safety and efficacy of Infliximab biosimilar CTP13 in patients with inflammatory bowel disease, specifically with ulcerative colitis and Crohn's disease. CTP13 became the first monoclonal antibody approved through the EMA's biosimilar regulatory pathway for rheumatoid arthritis, ankylosing spondylitis, adult and pediatric Crohn's disease, ulcerative colitis, psoriatic arthritis, and psoriasis. This study addresses the issue of confidence using Infliximab biosimilar in inflammatory bowel disease and switching from the originator. Concerns included different dosing of Infliximab, 5 milligrams per kilogram in IBD, as well as the use of concomitant immunosuppressive medication. Before beginning my analysis, let's hear about some of the study's highlights from the lead author's perspective. What do you feel are the three most important highlights or findings of this study? The most important summary points are 547 patients using CTP13, 97 of whom were switched from Infliximab, were followed for a mean of 4.3 plus or minus 2.8 months. The rate, 12.2%, and characteristics of serious adverse events, SAEs, was in line with previous experience with Infliximab. The occurrence of infusion reactions and drug withdrawal for SAEs was 7% and 8.2%, respectively. The whole efficacy in terms of induction in or maintenance of remission and response was approximately 90% at 24 weeks. The rate of primary failure was 8.1%, and loss of response, 18.6%, was in line with previous experience with Infliximab. What impact do you think this study will have on the utilization of the biosimilar Infliximab in patients with IBD? This experience has increased the confidence in the use of the first Infliximab biosimilar in real-world practice, although long-term data and more information after switch are awaited. This was a multi-center study from 31 referral centers studying patients previously diagnosed either as ulcerative colitis, 234 of them, or Crohn's disease, 313, to review the efficacy of biosimilar CTP13 on patients who received treatment for at least 8 weeks. The study included 311 patients who were naive to anti-tumor necrosis factor alpha agents, 139 who had a previous exposure to biologics, and the remaining 97 were switched to CTP13 after a mean of 18 plus or minus 14 infusions of Infliximab, reference product. Here were the study's key findings. After 2,061 infusions, 66 serious adverse events were reported, 12.1%. 38, 6.9% of them were infusion-related reactions. Infusion reactions were significantly more frequent in patients pre-exposed to Infliximab than to other anti-tumor necrosis factor alpha inhibitors. The incidence ratio is equal to 2.82 with a 95% confidence interval of 1.05 to 7.9. Efficacy in terms of induction in or maintenance of remission and response was high, with efficacy at 24 weeks of approximately 90%. For this specific study, efficacy estimations at weeks 8, 16, and 24 were as follows: 95.7%, 86.4%, and 73.7% for naive patients, 97.2%, 85.2%, and 62.2% for pre-exposed patients, and 94.5%, 90.8%, and 78.9% for switched patients. Although no direct comparison was performed, preliminary data on efficacy and safety of CTP13 were in line with those of Infliximab. Outcomes of the suggested studies all show CTP13 to be effective and safe treating IBD in real clinical practice. Here are my thoughts and analysis of this study. Biosimilars are designed to be similar to the originator biologics. biosimilars undergo a large battery of tests to demonstrate this similarity. The testing includes in vitro tests showing how similar the drugs are, including how well the drug binds to its target, and in vivo testing showing that the pharmacology of the biosimilar is similar to the originator. Given this similarity of the biosimilar to the innovator and the lack of magic in medicine, we should expect biosimilars to perform similarly to the innovator drug. The main finding of this study is that, as expected, the biosimilar gave similar clinical outcomes. This study is not going to suddenly change a lot of people's treatment outcomes, because even though the biosimilar works, patients who could be effectively treated with biosimilar Infliximab could be treated with the originator. I doubt restrictions on use imposed by payers would be changed by the availability of a biosimilar. This study may make doctors and patients more comfortable with the idea of using a biosimilar. Insurers may use these data to encourage greater use of biosimilars if doing so provides a cost savings. While this study does not rule out the possibility of small differences between the biologic and the innovator, we need to realize there may be small differences even between different batches of the innovator product. This study is not informative about whether today's innovator Infliximab works the same as the Infliximab that was used in clinical trials over a decade ago.